Okay, so we'll move on to my talk. So I'm going to be talking to you about an integrated approach to managing immunogenicity risk and drug immune modulation. So we've already heard some great introduction already to some of the challenges associated with characterizing immunogenicity of biologics. And Herman's already said that ultimately you've got to think about new proteins that are being developed for therapeutic use as containing potentially novel sequence content. And that can be a key reason for the immune system recognizing protein as non-self and so therefore reacting against it. But it's not that simple, because if it was then just an approach of eliminating T-cell epitopes or B-cell epitopes from those proteins would make life very much simpler than it actually is. And so understanding immunogenicity is a very complex area, multifaceted, and there are many different things to really consider in regards to what drives unwanted immune responses. So I've sort of put those into two different areas down there, both extrinsic and intrinsic factors that can explain or at least help us to understand immunogenicity. So extrinsic factors are things like the route of administration. So how is your therapeutic being delivered? Whether that's subcutaneous or IV and other routes as well. How frequently are you exposing the patient to this therapeutic? If it's a routine exposure, then obviously there's higher amounts of exposure, and therefore you can induce an unwanted immune response against that therapeutic, as opposed to a single dose where the risk is reduced. The dose of that protein, the patient's immune system status, are they immune suppressed, or is their threshold of T-cell activation and overall immune activation at a different level, which means they're much easier to tip that balance that Farnaz was talking about in an area that we don't want to achieve. Is there pre-existing anti-drug antibodies against the same therapeutic, or at least elements of that same therapeutic? And also, what's the HLA type of the patient? So there's many different factors. Intrinsic factors we have at least some control over, but not exclusively, such as the amino acid sequence of our protein, but also how it's formulated, and also the aggregation status of that protein. So higher amounts of aggregate can drive unwanted immune responses. So overall, thinking about immunogenicity risk from an earliest possible time point is what's really important, because if you can demonstrate that your therapeutic is at a lower risk than others, that certainly is quite a key advantage. Now, I'm not a big fan of cartoons because they can never do the immune system justice, so obviously it can only focus on a few points. But ultimately, what I'm talking about in this presentation is mainly thinking about this interaction between elements that are presented by the antigen presentation cell to the CD4 positive T helper cell. So ultimately, a biological or therapeutic can be taken up by an APC, naturally processed and presented, and peptides are displayed and presented on MHC class II. And it's these peptides that are presented to T-cell receptors on the CD4 T cells, and this interaction, of course, is very specific. And activation of the CD4 T cell goes on to induce proliferation of the cells and also production of cytokines. And these cytokines can go on and help our B cells become plasma cells and produce antibodies, which then bind to the target of interest, and these are the anti-drug antibodies that we're trying to stop happening. So because the T cells are a critical element in driving this immune downstream consequence, this is why many of the assays that I'm going to be talking to you about today are focusing on this interaction between T cells and the antigen presentation cells, so the peptide MHC interaction. But there are other elements as well in relation to innate immune responses, particularly cytokine storm, so I'm going to be thinking about that a little bit as well. Now, there's no single approach that people take to understand unwanted immune responses. But ultimately, we're thinking a little bit here about lead selection and optimization. So maybe you have a number of different variants that you want to characterize in more detail. You might want to think about the T-cell epitope content, because certainly the more T-cell epitopes that are present, then the higher risk there is of driving an unwanted immune response. You might be interested in characterizing completely novel scaffold proteins. There's a really exciting time at the moment with many, many different types of molecules being developed by many different organizations. Some of them are going to hold great promise, others maybe less so, but understanding how to select those different variants, one against the other, is very important. If you're generating junctional regions by binding two different proteins together, you can induce neoepitopes. So new T-cell epitopes can inadvertently be designed into a drug. So it's very important to understand what those risks are. You might want to assess formulation variants. Actually, how your product is being formulated can have a big impact on how the antigens are being displayed to the immune system as well. One area which I would say that we're still on the cusp of understanding is thinking about patient stratification and much more tailor-made medicines. Not necessarily of thinking about one therapeutic for one patient, although of course that is a strategy, but actually thinking about which molecule is most appropriate for different patients, depending on their immune status. So thinking about lead selection optimization is very important, but also immune monitoring, thinking about how we can track immune responses once a patient has received a therapeutic is also really important to help us understand where the immune responses are being generated and how we can intervene at the appropriate moment. So this approach of thinking about choosing different leads, how to track those immune responses, can really overall strengthen your regulatory position. If you can understand what's going on at a deeper level, that supports this quality by design strategy, which the regulators, both in Europe, both in the US and globally, are very, very keen to see. And they're learning as well. We're all learning together, but they want to try and understand how these tools can help us and inform us to make the right choices. So anti-TNF is a huge market. These antibodies and other molecules against TNF-alpha are a multi-billion-dollar market globally. And so we're talking about molecules such as Humira, adalimumab, which is a fully human, generated by human phage display technology, antibody against TNF-alpha. Now, despite its promise, it turns out it's now recognized that about 50% of patients who are receiving Humira, which was the kind of the best-in-class drug, actually develop neutralizing antibodies against the antibody itself. And so patients then have to switch to a different anti-TNF-alpha antibody after 18 months because of the treatment failure. And so, yes, you can maybe switch and keep trying a new different antibody, but that's a real expense to the healthcare system to try and identify which molecule to use. And of course, it's very disruptive to the patients who have enough to deal with as well. So what we wanted to do, and I'm just using this as a case study, is to sort of show you how we can identify and characterize immunogenic epitopes of adalimumab. So the first question in this sort of COGS are thinking about antigen processing. And we're asking the question, which peptides from adalimumab are naturally processed and presented to T cells by MHC? So to do this, at ProImmune, we have a biobank. Here in the UK, we're regulated by the Human Tissue Authority. And we source donors from NHS Blood and Transplant. The donors are then, the PBMCs from those donors are isolated and cryopreserved, and the full HLA type is carried out. So we can actually pull donors out of our biobank to either give us a broad global distribution of MHC types, or we can narrowly focus on a particular MHC haplotype if that's of interest. And these cells are deployed in many of the different assays that I'm going to be talking about today, but not all of them. I'll highlight where it's different. And I'm going to talk to you now about Propresent, which is an antigen presentation assay. And Propresent is a tool which allows us to sequence peptides that are naturally processed and presented by the MHC molecule using a tandem mass spectrometry. So it allows us to think about how the drug is actually seen by the immune system. It may be that we're spending a lot of resource and effort in trying to manipulate a particular sequence because we think it's a risk. But if that element of the protein isn't actually ever going to be shown by the immune system or is a very low risk of being displayed, then maybe we need to shift our priorities a little bit. So it helps us to understand a little bit more about the mechanisms of how our drugs or other molecules are presented by the immune system. We can evaluate the impact of modifications on proteins, so things like post-translational modifications, glycosylation, for example. How does the presence or absence of those glycosylation patterns alter peptides that are presented? It's an area which we're still becoming, we're not very clear about, but we're becoming clearer. We can also use this tool to think about vaccine developments. So rather than unwanted immune responses, we can use this to identify completely new targets for developing new vaccines. We can take a whole organism, we can put it into these cells, and we can identify which proteins, which specific peptides, but from which proteins these peptides are from. It's a very powerful technique to identify what's going on. Now, I'm going to be focusing on data looking at MHC class 2, but we also have tools for looking at MHC class 1. And it's a very quick tool. We can look at typically a panel of 10 to 20 donors in around about 6 weeks. And we have a number of publications as well, which are now in the literature. So the first thing that we do is we take the protein sequence of interest. So we need to know your, for example, antibody sequence. So here we take Humira. We take the sequence, we load it into a database. And the database is the entire human proteome. And on top of that, we take the sequence from adalimumab. and we're generating mass spectra from all the different peptides. I'm just going to switch to the next slide just to show you how it works. So protein of interest is loaded into immature DCs. These dendritic cells are then matured, and the protein is naturally processed, and peptides from that protein are then in the context of class 2. We then lyse the dendritic cells, and these DCs with the class 2 on the surface and the peptide are immunoprecipitated. So using an antibody, we can pull down different classes of MHC. So we can pull down, for example, just HLA-DR. We can look at DP, we can look at DQ, or we can look at MHC class 1. The peptide's then isolated from the groove of the MHC and then run through a very sensitive LC tandem mass spec instrument to then identify the whole range of peptides that are present. Now, the MHC's job is to process and present peptides from your range of different proteins. So typically, you'll see peptides that are from many thousands of different proteins, and we're talking about tens of thousands of peptides overall. But you're not interested in those peptides. You just want to know which peptides from adalimumab are being presented. So we carry out a number of different QC steps that I've mentioned. So we generate a decoy database, which is the whole protein sequence and your protein of interest. And we scramble it to make sure that all the hits are specific. We also have obviously standard database, which we're probing to identify all the hit peptides. And we have a 95% confidence-based exclusion search. Most of the peptides that we identify are typically a range of lengths from 11 to 17, 18 amino acids in length. And that's, of course, in line with what you would expect for MHC class 2. And we also have a number of QC steps which allows us to identify peptides from a set of control housekeeping proteins. So if the donors are not presenting peptides from these proteins, then they're excluded from the analysis. We want to make sure that it's working as it should. And what we can do, this is just a subset of the data, so I mean it's not too important, the absolute details here, but donor by donor, we see the HLA type, and we see the specific sequences that are presented. Some donors you might not see any peptides from Humira. Some donors you might see a single sequence or two sequences from the same portion. Other donors you might, for example, this one here, we see three different regions identified, but overall 3 sets of nested peptides. This donor is a large region, a large nested set. And that's just an example of the kind of data that you can generate from this tool. But if we summarize it, then what we can see is that there are actually 4 different regions that are presented, all from the CDRs from Humira. Now that's not too surprising because these are the kind of the most important immunogenic regions. But even then, we have much more information. It turns out that this red sequence, for example, out of 20 donors that we analysed, was only present in a single donor that was DR1 and DR0403. So, fine, that's interesting, 1 donor out of 20, but how relevant is that if we're actually just trying to look at the bigger picture? Whereas if we look at this blue sequence, this was actually present in MHC types that are listed here, but actually in half of the donors that we analysed, this particular peptide or a region of this peptide was present. So this shows us that it's a really promiscuously presented peptide and therefore much more likely to be a relevant T-cell response. I'll come back to that in a moment because hold that thought. But thinking about antigen presentation and how we've generated this data, we also have a database which we call Presentium. And rather than thinking about exogenous proteins that have been loaded into the dendritic cell, if you're interested in self-proteins and any proteins related to autoimmune disease, for example, then Presentium is a database that we've generated from many, many donors that we've analysed. And we can then identify specific peptides, obviously, that are presented from self-proteins. So all we need to know from our customers is a Swiss Prot ID number, and we can mine the data for you and provide you with a summary report, free of charge, so you can see how many hits we've got, if any. And if we have got hits, then we will provide you a full proposal, and then you could license that data from us. So this is what Presentium is. It's generated experimentally by ProImmune using a large panel of donors. These are all healthy donors, I should point out. They're not allergic donors. But it's a tool which is very, very useful for being able to understand self-proteins. And as an example of that, we've got some data here for alpha-galactosidase. The deficiency of this enzyme causes Fabry disease. And so here we actually find, looking at a large panel of donors, that we identified 49 donors that have significant hits. And it turns out that there are 25 presented sequences, of which 8 were unique. And of our donors, we have 3 homozygous donors, so we even know the specific HLA type that these peptides are presented on. And so that's the kind of the level of detail that you can have, and obviously if you license that information, you'll have the specific sequences. I'll forward wind to that. Okay, so just because a peptide is presented by MHC doesn't mean that it's functionally relevant. So it's really important to not just think about one of these cogs, but you have to think about all of them. So T cell function is the next question. Do presented peptides that we've seen here from adalimumab elicit a functional T cell response? So here I want to introduce our ProMap CFSE T cell proliferation assays. These are very routinely used by many different groups around the world, and we'll probably hear a little bit more from Bernard later, maybe a slightly different method. But ultimately, this particular assay we're using to map the functional relevance of these peptides that we've identified. So we're using PBMCs from typically 40 or 50 donors, in this case 40 donors, and the cells are depleted of CD8s, and they're all tissue typed. And what we do is we either generate overlapping peptides from your protein or your region of interest, or we can use the peptides that we've identified from this antigen presentation assay. And we do a 7-day culture in a CFSE proliferation assay. For those of you who aren't familiar with this approach, it's very simple. It's a flow cytometry assay where we take PBMCs and we label them with a fluorescent dye CFSE. We're stimulating the cells with peptide, and as you get proliferation, the dye is then divided equally between the daughter cells, and so you get reduction of intensity of your proliferating cells. So your plots, you can see your cells in the top right quadrant, they're labeled as CD4 positive and CFSE positive, and then when they proliferate, they move to this upper left quadrant. You'll have some natural turnover, but when you have a strong response, you'll see a population in this upper left quadrant. So this map here is a colourful sort of interpretation of the data, but basically every different colour is a different donor that has responded significantly to a specific peptide. So these are all overlapping peptides from Humira, and you can see, for example, in this region, in the middle of CDR3, we see one peptide in which 7 out of 40 donors have responded significantly, comparing it to our controls. Interestingly, though, our peptide of interest, the one that we've actually shown to be presented promiscuously by these healthy donors, is actually also a functionally relevant peptide. Not the highest one, but nevertheless a significant response. And so now we know that this particular peptide, not only is it presented by MHC, but it also, in a natural situation, but also there's a strong T cell response generated to it. So it's a key T cell epitope in adalimumab. And it turns out from other discussions that we've been party to that in the public forum that we know that this particular epitope is a key region of interest for the developers of Humira, and they're thinking about that for their next generation. But what if you're not interested in the individual peptides? Maybe you've got a number of different variants, and you want to think about how do I choose one variant from another variant, but still think about functional T cell responses. Well, this is where our ProStorm dendritic cell T cell assays come in. So it's a whole protein analysis. And like in the antigen presentation assays, we're generating dendritic cells, monocyte-derived dendritic cells from panels of healthy donors. And these DCs are then loaded with different antigens. It could be a panel of up to 18 different proteins, different antibodies, for example. And the protein is naturally processed and presented by the dendritic cell, so you take into account post-translational modifications, aggregation, all those other factors. And the peptides from your protein are then presented to autologous CFSE-labeled PBMCs. So these are T cells from the same donor that the dendritic cells have been derived from, so there's no allogeneic interaction at all, and allogeneic stimulation. So all the T cell responses that we see are really driven by the protein that's been loaded into the cells. And so then we measure T cell proliferation after 7 days following incubation with a test article. So what we generate is an antigenicity index, we call it a response index, and this is a measure of not only the percentage of donors that are responding significantly, but the strength of that response as well. So that's really important because in a dendritic cell, you're really taking the power of the dynamic range of these assays. They're very potent stimulators, and so you really want to take into account not just whether the T cells are responding, but actually how strong is that response as well. So in this way, we have our controls, but it allows you to then comparatively measure the responses of your different antibody therapeutics. That's typically what's being used, but novel scaffold proteins, things like that. These aren't absolute numbers, these response indices, but they are qualitative, I would say, because ultimately if you use a different 40 donor, 50 donor panel, you will have a slight variation. And obviously you're limited by the number of cells that are available from a single donor. But ultimately we see that there's the same pattern emerges from how you analyze these different proteins. So the other thing I wanted to bring up was how the peptides that we've identified in these assays, what's the HLA restriction got to do with it, and is that important? I won't spend too much time going over this, so I will just bring up a picture. We have recombinant MHC peptide binding assays. Now, a lot of people who are thinking about immunogenicity, particularly maybe 10 years ago, were thinking about using algorithms purely to predict immunogenicity. Now, that was a bit of a mistake because MHC peptide binding is just one piece of the picture. It's an important piece. You have to have peptide binding, of course, to then induce a T-cell response. But if you're just trying to predict which peptides bind, well, everything's going to light up and it's going to be very over-predictive. Now, in silico tools have their place. They're very important because if you have huge amounts of sequence to characterize, where do you start? You have to start somewhere. But ultimately, things are improving, but we may hear a little bit more about that later. But it's still quite a challenge to try and understand exactly how peptides are predicted to bind to different MHC molecules. And so we have, and we've been doing this for many years now, a physical MHC peptide binding assay called REVEAL. And we're using recombinant MHC molecules and incubating peptide. And the peptide binds to MHC, and when it does, we have a conformational specific antibody, which will only recognize correctly conformed MHC peptide class II complexes. And so you measure signal in an immunoassay. Now, there are different elements to this, not just binding, but also stability, because if you imagine it's a very dynamic process, a peptide binding in the groove of an MHC molecule could actually, once it's bound, can fall out quite quickly, but once it's bound, it could also stay stably bound. And it's how long a peptide is actually in the groove and is presenting to a T cell, which increases its chance of being a functionally relevant peptide. If a peptide just falls out, it's not going to be there. So we do a stability assay as well, and that's important to take into account how likely those peptides are going to be remaining in the groove. So I'm going to combine the three different data things experiments I've been talking about, the T-cell assays, the antigen presentation assays, and the MHC peptide binding assays. So bear with me, I'll try and do this hopefully in a clear way. What we're looking at is just one peptide. This peptide 102 is the one that we saw functionally relevant in the T-cell proliferation assays. These were the 5 donors that responded significantly to that peptide, and listed in this table is the HLA types of that donor. So donor 1 is DR 1301, 1501, DQB 0602, 0603, etc. Now, to make life a little bit simpler, if you remember with the antigen presentation assay, we were using an anti-DR antibody to precipitate down. So because we're not interested in DQ and DP for this set of data, I'm just going to hide it so our brains can relax a little bit. Now, the colours relate to a sort of a traffic light scheme. So red is no binding in the physical MHC binding assays. Green is strong binding. Yellow is weak binding. And grey is not tested because that allele wasn't in our panel. Now, we have 56 different alleles in our panel, so they are pretty extensive, but of course it's not every single one. So great, we can see that these donors have responded physically in the functional T-cell assay, but this is one peptide. How does this sort of tie in with the antigen presentation assay? Well, if we look at this third peptide, which I should have helpfully coloured blue, which is what we saw before, but this is the blue peptide that was responding in 10 out of the 20 donors. This donor is DR1, and so the donors that responded in this assay and were presenting the peptide were DR1 and DR0301, amongst others. And you can see that it's DRB1 and DRB0301, which this peptide also binds to. So the state of the art at the moment is that we can combine all this information together, and we can say that if you are a TNF-alpha or patient receiving therapeutic levels of anti-TNF-alpha antibody, and you are DRB101 and DRB0301, for example, then we know the specific epitope that you should track those immune responses to. Now that might mean, and I'm not saying that it is, that if you are DR1, DR3, then you shouldn't receive Humira. It's going to be more complicated than that. But at least it's a sort of a counter-indication to think, well, actually, maybe you should be having one of these other anti-TNF-alpha drugs, for example. It is going to be more complicated, but it's just one of the ways to sort of think about it and understand the level of detail at which we can go to try and characterize therapeutics. Probably going to hear a little bit more from Paul a little bit later relating to class 2, but I also wanted just to highlight, to track these immune responses in patients, what people haven't done yet is to use class 2 tetramers to actually look at these T-cell responses in patients. And that's now possible. And we haven't got data to show you measuring in these patients. That would be very nice, but we haven't got there yet. But I just wanted to highlight that we have these reagents now for tracking antigen-specific CD4s. And that is very commonly used in viral research, viral antigen-specific research, and cancer as well, but less so in the area of unwanted immunogenicity. So I wanted to just bring that up as a thought so that you can know that you can get them from us if you need to. And then we have multiple different donors. This is, again, just looking at different examples of tetramer staining, in this case in HCV. And you can see here, these are just 2 different epitopes, and this is stained from data from Gail Glau's group over at Mass General in Harvard. So the last thing I'm going to talk about in the last few minutes is really just an assay which we call ProStorm, cytokine release assay. Now, cytokine release is a first infusion reaction which can occur in response to biotherapeutic administration. It can be very, very rapid, and there are many different forms that it can take. But ultimately, the range of symptoms can be quite mild, ranging all the way through to fatal. So you have to be pretty careful with it. And the worst example was this T-Genero product, which was an anti-CD28 antibody, and it caused very unanticipated and severe reactions in all the test subjects that received that in North London. And these weren't predicted by animal models. So people want to have better ways at trying to predict what might happen before you get into the patients. So the regulators have now come forth and provided some guidance in this area, and they say that in their immunogenicity assessment for therapeutic protein products, data from animal studies should be supplemented by in vitro assessment of cellular activation, including proliferation and cytokine release in human whole blood or PBMCs, and that measurement of a cytokine panel should be as broad as possible and definitely include IL-6, interferon gamma, and TNF-alpha. So what we've done is we've developed ProStorm as an unmanipulated and undiluted fresh whole blood assay. So this assay does not use our cryopreserved donors. In this assay, we recruit donors directly for each study. And we have 50 mils of blood from each donor. Obviously, consent is obtained, and most studies are usually between 20 or 30 different donors. What we're doing is we're adding in test articles at a range of different concentrations to the whole blood, and then literally measuring within a few hours. The blood is in the lab and we're doing the incubation, and then there's an overnight incubation step, and we then harvest the plasma, and we measure the quantities of the specific cytokines present in that plasma. So we have an assay negative control, which is PBS, and staph enterotoxin B is our positive control. And we typically run Erbitux and Campath as low and high response comparators. So Erbitux tends to give a pretty low response, whereas alemtuzumab is a high response comparator. And what we're doing is we're measuring a whole panel of different cytokines, not just the ones that the FDA focus on, but additional ones as well. So just to bring you into line how we're presenting the data, or one of the formats that we're presenting the data, these are the 4 drug concentrations at the top, so 0.1 up to 100 micrograms per mil. And along the bottom, we have a logarithmic scale between 1 and 100,000 picograms per mil for the cytokine. This is IL-6. IL-6 is probably the cytokine which is most associated with cytokine storm, and therefore I'm using it as an example here. And in this case, we're incubating the cells with Remicade. Now, each diamond is a different donor. And as the donor, as the dose increases, you can see that there are donors that then become responders, and they're filled in diamonds at that point. And this vertical red line is a threshold. It's an arbitrary threshold, which is based on Erbitux, our low response comparator. So as soon as you have a donor which kind of goes over this line, then it's nudged into the positive filled-in version. Now this is, I apologize, it is quite small, but I'll just highlight, first of all, the positive control, which is the top right. So Campath, we can see that as you increase from 0.1 up to 100 micrograms per mil, pretty much all the donors become positive. So that's actually 27 out of 29 donors in this case are positive at the highest concentration of Campath. But even at 1 microgram per mil, we have 22 out of 29 donors positive. So that's our positive comparator, or high response comparator. Erbitux, on the other hand, you can see the majority of donors are actually on the baseline, but there are a few donors, and in this case, and even the highest concentration, it's still just one. It's always going to be 1 out of 29 in this case that's positive, because we use this as a 95% threshold limiting factor. But there is only 4 other donors which are just below the line here at the highest concentration. And so what it allows you to do is to benchmark where your molecule lies in the overall scale of things. So if you aggregate, if you take Remicade, you can see that as you increase the concentration of Remicade for IL-6, then the percentage of donors that are responding comes up. If you aggregate Remicade by stir stress, that really switches on the immune system. And not surprisingly, really, but all the donors, or 27 again out of 29 of the donors, are very strong responders there to aggregated protein. So just to give you an idea of how you can then rank your protein against others in this sort of benchmark. And it's useful because it's never going to predict what happens in the clinic. There are always going to be other factors, but it helps you to understand what that relative risk is, and that's really important. And so we do this very quickly. Most studies are turned around within about 6 weeks from start. So just to summarize, hopefully I've managed to inform you and discuss that managing drug immunogenicity by design is really important at thinking about it from an early stage as possible and to win the drug markets in the future. Generating this data allows you to differentiate your biologic from the competitors, and really you do need access to a wide range of specialist assays. One assay is never going to give you all the answers, so getting a broad understanding is very important. And our breadth of experience and integrating these different assays is really key to reducing the overall risks of projects and getting things moving forward quickly. We have a lot of experience. I've been talking about antibodies predominantly, but we have a lot of experience in the variety of different assays that we've worked in, using bispecifics, biosimilars, antibody-drug conjugates, vaccines, therapeutic peptides, all sorts of different scaffold proteins, orphan drugs, enzymes, and replacement factors. So there's pretty much most classes of molecule for this particular sort of set of assays. So again, thank you very much. If you haven't had a chance to wander into town, I do recommend it. I always like to say when I show this slide in other parts of the world that that's my office there, but of course, you will know better. But thank you very much, and I'm very happy to take questions.